So I'm going to be recording this lecture. Can you hear me?
Yeah.
Okay, so we are starting with chapter 2.3. And
Teacher.
Yes.
Teacher, the
Yes.
handout that you give us is 2.4.
Yeah, you don't need this one. This is just a little bit of exploration. I will.
Okay.
2.4, yeah. So this is explore application of cell biology and nanotechnology in medicine. The biggest breakthroughs in medicine may soon come in the smallest packages. So nanotechnology is the study of material in nanoscale, or nanometer is 1/1000th of a meter. So human hair is 10,000 nanometer wide, and a sheet of paper is about 100,000 nanometers. You can get an idea, a nanometer, how small it is. So this is a picture of an artist illustration of nano robots attacking red blood cells. Okay, so this is what the cell biology is going towards, a nanotechnology, and having the gene scientists are researching ways to use nano-size robots or nanobots in areas such as cancer research, cell and bone repair, and gene therapy and drug delivery. So that was just chapter 2.3, but we are going to start chapter 2.4. And I already put the handout for chapter 12.4, as well as I put the questions in the handout. Now, let's start with the topic of transport across membrane. Chapter 2.4. Transport across membrane. Now, for a cell to survive a function, it must take in nutrient and expel waste, and communicate with its environment and neighboring cells. The exchanging of substances is a complex process because plasma membrane is highly selective. So it must be able to take large molecules while prevent small molecules to enter or leave the cell. It must be able to recognize foreign substances and that are harmful and block their passage. So expelling the toxic products. So further complicated matters, some molecules cannot be stopped. The plasma membrane, they enter and exit regardless whether it is not or it is desirable for the cell. So when this happens, the cell must withstand the consequences. So the transmembrane exchange of material is not limited to the outer surface of a cell. Countless substances must be able to cross the organelle membrane with eukaryotic cells. So in mitochondria and chloroplasts, chemical reaction take place within the internal fluid. So I'll write this down, and so it will help us remember. So mitochondria, and we studied what is mitochondria last week. Mitochondria and chloroplast. Reaction takes place in the internal fluids. And chemical reaction separated from cytosol. From cytosol by 2 or 3 membrane layers. So for this reaction to occur, reactant must enter to the organelle while products are being permitted to leave. In this section, we will look into free movement of certain molecules, such as, so we will look into movement of some molecules such as oxygen, carbon dioxide across membrane. Okay, as well as in a specialized transport mechanism that are used to control the entry and exit of the molecules. So let's look into, first of all, for a new definition. So definition of passive transport. So the movement of a substance across membrane without expending energy. Okay, now that is the passive transport. So diffusion drives the passive transfer. So diffusion drives the passive transport. So now diffusion is the net movement of a substance from is a net movement. I'll just change. Is a net movement of the substance from higher concentration. So from higher concentration to a region with lower concentration. Okay, so it occurs because molecules are in constant motion and in an ideal closed environment tend to become uniformly distributed in a space. Diffusion primary is a mechanism of a solute movement within a cell and between cellular compartments separated by membrane. If molecules are more concentrated in one region of a solution on this one side of the membrane, the random motion of the molecule causes them to become evenly distributed. So I'll show you a picture that will help us understand this. So if you look here, this is figure 1 from your book. And a diffusion proceeds, there's a net movement in one direction until the concentration of both sides of the membrane become equal. So let's look at this picture here. And this is water, and I have molecules here. there's a net diffusion this side. So what it wants to do, the molecules want to move to this side because there's less molecules there. So there is a net diffusion. Again, they are moving across the membrane here. This is the membrane. And now this, what happens is that now I have my molecules even distribution. So I have something called equilibrium here. Okay, so what happened here? My molecules moved from higher concentration to a lower region with a lower concentration. So the rate of diffusion, now the rate of diffusion, the rate of diffusion depends on the concentration difference. Depends on the concentration difference. Okay. So, or concentration gradient. So that also the word is, I'll write it here, concentration gradient. Okay. The faster the rate of diffusion is, even after concentration of molecule or ion is the same in the both region, molecules or ion continue to move from one region to another. However, there is no net change in the concentration. And this is an example of a dynamic the equilibrium. So we have a new definition called dynamic equilibrium. Okay, and is the state in which continuous action, results in a balanced condition. So membranes have selective permeabilities, which means that some molecules can diffuse rapidly across the membrane, while other molecules are unable to transit without a membrane assistant. So two major factors are size and charge. So I'll write it down. Membrane can be selective permeability, selective permeability, meaning some diffuse rapidly while others are unable to transit across the membrane. And there are two factors. One is size. These are the factors play a role, as well as the charge of the determine each ease of the molecule or ion can move across the membrane. So there are two types of passive transport. So remember, we looked into passive transport here, right? Passive transport. So now passive transport is divided into two. Passive transport, okay, is divided to two. One is simple diffusion, and another one is facellated diffusion. So facellated diffusion. Okay, so let's look into what is a simple diffusion. So this one, definition of the simple diffusion. Okay, now in simple diffusion, the ability of small and non-polar substances move across the membrane as move across the membrane unassisted. Okay, so that is simple diffusion. So let's look in a picture to help us kind of understand what is a simple diffusion. Okay, so let's look at this picture here. This so in the definition, first of all, it said small and non-polar substances move across the membrane. So let's look here. This is a picture of a membrane here. And I have oxygen, carbon dioxide, and they are soluble in hydrophobic interior of the membrane and move from one side to another side. So the oxygen move. Now next in the definition, that was my small molecule. Then it talks about non-polar molecules. So non-polar steroid hormones or non-polar drugs can actually move easily like this. So glycerol here, we have a small uncharged polar molecule. They move. Non-polar drugs, they can easily move from one side to another. So that is a simple diffusion. Now we have water, glycerol, we have polar. These polar molecules can still move through the membrane and rapidly. And we have impermeable to large molecules. So this one, you see these large molecule here or ions here. You notice they are not able to move across the membrane. They don't. So compare the rate of a transport of water, the movement of these small ions is about 1/1000th of a speed. Okay, so that is about simple diffusion. Then next, we had a definition. Again, we had here passive transport. We talked about simple diffusion. And next, we are going to talk about facellated diffusion. So let's look into what is a facellated diffusion. So facellated diffusion. Okay, so facellated diffusion, a slow rate of diffusion, may not keep up with the demand that metabolic process often have ions or polar charged molecule. So the facellated transport of ions and polar molecules through a membrane via something called protein complexes. Okay, so let's look, what does that mean? So let's look here. I'll show you a picture, and this will help you kind of understand what I mean by phasylated diffusion. So if you see here, we have plasma membrane here. These are my membranes here. And I have a little protein here, protein complex. And this kind of makes it easier for phasylates the transportation across the membrane. So across the membrane, it helps to phasylate by the protein complex. This is known as phasylated diffusion. So although phasylated diffusion involves a specific transporters, the movement of molecules and ions is still driven by diffusion based on a concentration gradient across the membrane. So when equilibrium is reached, there is no longer a concentration gradient. Phasylated diffusion stops. And phasylated diffusion is carried out with the internal membrane protein called transport protein. Again, is this one here. And the extent throughout the membrane, there are two types of transport protein. And so let's look into that. So transport protein, there are two types of it. So one is channel protein, and another one is carrier protein. Okay, so channel protein is hydrophilic pathway. In the membrane through the water with certain ions can pass. And other channel proteins facilitate transport of ions such as sodium, potassium, and calcium, and chlorine. These are the ions, and the most ion channels occur in eukaryotes, and they are voltage gated. So let's look into the carrier protein, and we will go on a break after this. Carrier protein is a protein that binds to a molecule and transported across the lipid bilayers. So this binds to a molecule and transports it across the lipid bilayer. Now, I want us to go on a break, and I will come back in 5 minutes. I will put a new meeting. Do you have any questions so far?
No.
Okay, so I will bring back put a new meeting in 5 minutes. So we go on a break and we continue with